Right, so today I'm going to be talking about some of the work going on at the Allen Institute toward trying to characterize different components of cortical circuits using a high throughput combined experimental and computational approach. So what I do at the Institute is focused really on building models of individual neurons at kind of a biophysical level, but I'm also going to talk a lot about the data generation pipeline that's going into generating these data sets and that goes into the models, as well as kind of how this whole effort fits into the broader Institute goals. So just to give a quick overview of what the Institute is, for those of you who may not know. So we're an independent nonprofit research organization. We were founded in 2003 and focusing on the brain sciences. And so we've actually been growing pretty rapidly over the past few years. We've added over 100 people from 2011 to 2014, and we're actually projected to be close to 500 by 2021. Last year our budget was around $60 million per year, and basically this has all been made possible by the generosity of our founder, Paul Allen. Our culture is kind of a mix between a university and a biotech startup, so we really try to focus on these larger projects that are done at scale, but that also require a lot of interaction between different types of people working with different techniques and in different disciplines. So we're all focused on the main goals of the Institute, but working at different levels, but very collaboratively. And actually, so kind of historically, the Institute's been known for these brain atlases that have been made available to the public online over the past 10 years. But actually in 2012, we started this new program for building what we're calling cellular level observatories for mouse and human cortex. So those are, I'll talk a little bit more about that, what that means, but kind of mouse and human are the species we're focusing on here. And in mouse in particular, we're actually focusing on the visual cortex is what we'd like to really investigate in detail. So as I mentioned, the Allen Institute has generated these online public resources. They're available on our website, brainmap.org. Most of these are basically, as I mentioned, atlases. But they're widely used by the community, and they kind of represent our philosophy of trying to be as open and sharing as possible. So all these data are publicly accessible via API basically as soon as they pass quality control. So we get them out there pretty much as quickly as we can produce it. And typically these data have been available 1 to 2 years before any publications from the Institute that use those data. So really we're not trying to keep things for ourselves. We want to share it as much as possible. And that's going to go for what I'm going to be talking about that we're doing that's kind of in a new direction apart from the atlases. So I mentioned this 10-year program, which we call Mindscope, and this is around kind of these cortical observatories, which we're thinking of kind of different platforms using different methodologies as ways of investigating a cortical function. And one framework that we use to break this down is kind of along these 3 different axes for understanding a complex biological system like the cortex. So in one direction we have components, so really kind of defining the parts list. This is something that Sebastian Sung talked about yesterday. What are really the cell types in the cortical circuit? What are the properties and types of synapses available? And then what are the basic circuits that these different cells form with each other? And then another dimension is computation. So really, what are the basic operations being performed by these circuits? So what is the sensory representation in the cortex? And then in another dimension is cognition. So what are the tasks that these circuits are performing using these computations? And the things we're going to look at are like object recognition, but then attention, decision, memory. Because obviously this is a pretty broad area, even though we're focusing on this relatively small area of cortex, we have to use a lot of different techniques. And so, things people are working on at the Institute range from, mice doing visual behaviors to, 2-photon population calcium imaging, like what, Jeremy showed earlier today, to, down to the scale of serial EM reconstruction and array tomography, and lots of things in between. So what I'm going to talk about is more along the components axis. It's kind of one of the first things that will be publicly available from this new 10-year program. And this is really focused around the question of cell types. So, the motivating questions for this line of work are, what are the cell types in the visual cortex? And then, similarly to what's been mentioned by several people, what are their physiological, morphological, and genetic characteristics? How do they overlap, and how do those really interact to define these cell types? And then, once we kind of have that list, the idea is to try to figure out how this cell type diversity is necessary for cortical computations. Like, is it necessary? And then, what roles might these different cell types be playing in the computation? So the platform that we're using to investigate this is something we're calling the in vitro single cell characterization pipeline. So this is based around having, kind of in a scaled way, teams of people who are focused on making single cell, whole cell patch clamp recordings from genetically labeled cells from the mouse visual cortex. So we have, a team of technicians who are making these recordings. There's a team that supplies them with those slices. There's a team that takes that recorded tissue and processes it for imaging, that kind of thing. And so we can do a lot of different types of modalities to generate a large database around these specifically identified cell types. And I'll just quickly go through that. So, first we're recording a lot of metadata about what we're actually recording from. So not just the Cree line that was used to label the cells, but also, where exactly in the brain was that. And we can map that to, a 3D common coordinate system that we're developing, through our atlasing efforts. So you'll be able to know where, originally in the whole 3D structure of the brain, the cell that was recorded from was located. We're also extensively characterizing these, since we're doing these patch clamp recordings through a variety of electrophysiological protocols that I'll talk about in a second. The cells are also being filled and then imaged so we can look at their morphology and then also reconstruct it. So we'll have a 3D representation of that morphology. In addition, there's another group that's pursuing a transcriptomic approach with the same Cree lines. So not, from the same cell as of yet, although that's something we'd like to do in the future, but at least we'll be able to see kind of what the cell type diversity is using a transcriptomic approach and then overlay that on the diversity that we're seeing coming out from the physiology and from the morphology. And then we'll also, the thing that I'm working on is building models using these data. So for a given cell where we have the physiology and morphology, we're creating a compartmental biophysically detailed model of that, as well as actually several different types of point neuron models for that same cell. So we'll have models of different levels of abstraction. We'll be able to kind of combine all this data to develop this data-driven taxonomy of cell types. And again, all of this is going to be made publicly available on our website, so we'll have this database that people can look at. So you'll be able to, on the web, look at a cell's physiological properties, see its 3D morphology, and download a model that, in some sense replicates its behavior. So as I mentioned, we're using Cree lines that have been characterized at the Institute as well as elsewhere to serve as kind of a genetic handle for defining specific subpopulations of cells that we can investigate using a variety of techniques, including the ones I just went through, but also things like the two-photon calcium imaging and that sort of thing that's going on elsewhere at the Institute. So we're using a common set of Cree lines and ones that label cells in kind of what we hope is an informative way. So, you can see at the top there's several different excitatory neuron lines that we're using. Many of these label cells in a layer-specific way, which we think will be useful. Some of them, we have multiple lines for labeling different populations of cells in different layers. And then with the inhibitory neurons, those tend to be less kind of layer-specific, but map to the broad categories of cells that many other people have defined, including like the parvalbumin-expressing cells, somatostatin cells, VIP interneurons, as well as others. So again, we're targeting these Cree-labeled cells, making recordings. And then the people who are doing the recordings are using this standardized electrophysiological protocol, so we'll be able to, in a very systematic way, compare cells of different cell types from the cortex. So all of them are going to be, all this data is going to be collected in basically the same way, with the same, so we'll get the same types of information. And here's just a few examples of protocols that we're using to characterize the cells. So this is not the full set, but it shows, we're using short, strong current injection to get at things like the instantaneous threshold, slow ramps, long current steps to characterize subthreshold responses as well as the superthreshold responses, and then noisy stimuli to get responses with a lot of frequencies going on at once. And then, as I mentioned, we're reconstructing these morphologies, so it's a little bit, I guess, hard to see on the right, but basically we have the stained image of the biosite filled cell on the left, and then using software developed by people at the Institute, we can enhance that image and then generate an auto-traced reconstruction of that cell, which is then taken by a team of annotators to basically validate that, correct any errors that might have come out of the auto-trace, and then produce the final morphology that will be published on the web. So as I mentioned, what I'm doing is kind of taking all this information and then trying to create models based on that to replicate their behavior and then use that as the basis actually for network models that we'll then be able to compare to other data being collected at the Institute. We're actually developing models at different levels of abstraction, which we think will be useful. So, on the left is what I'm working on, which are the biophysically detailed models. We're also developing point neuron models, as I mentioned, which are basically generalized leaky integrate and fire neurons. And then we have other people working on more of a population statistics model where you're not simulating an individual cell's firing, but rather describing the ensemble activity of a population of cells. So we think that this will be useful for both, obviously these different types of models have different computational requirements, so we can use them for different scales of neural network models, but we can also create even hybrid models, maybe have a small set of biophysically detailed neurons surrounded by point neurons with kind of long-range connections coming from a population statistics model. So that's something we'd like to drive toward as we continue to collect this data. So just to describe kind of the one on the left in a bit more detail. So, we're creating these biophysical models of single cells. What I mean by that is that we're taking this electrophysiology data and the morphology recorded from the same cell in the slice. And then we're putting around 10 active conductances at the soma and axon to generate its spiking activity. The dendrites in this case are passive. That's because we're kind of limited by the data that we have. These are generated from somatic recordings with a single electrode injecting current at the soma. So we know that there are active conductances in the dendrites, and we have people working on those types of models, but we're a little bit data constrained kind of at this point. So basically the parameters of the model are the densities of these active conductances, which we then vary and optimize using a genetic algorithm based on methods developed in Idan Segev's lab. And then we're assessing the fitness of the model by comparing the electrophysiological responses or features that we calculate from those responses to the variability of those features within the single cell, the experiment that we've recorded. So as you can see kind of from the example at the top, we can get models that do a pretty good job of reflecting kind of the global features, I guess, of the cell. So things like the overall firing frequency, the action potential height. You can also see from the example that we're not completely consistent with the action potential shape. That may or may not matter at the scale that we're looking at, but that's something we're actually investigating. And then on the right, you can just see kind of the movie of the cell spiking in response to a current injection. You can see the change in membrane potential is biggest at the soma, and the decays out again through the passive dendrites, as you'd basically expect. Just to kind of to clarify what features we're talking about that we use for the fitting. So we use a set of kind of standard single action potential features, things like the height, the width of the action potential, the voltage where the threshold is, the depth of the after hyperpolarization, that kind of thing, as well as these firing pattern features from the response to these long current steps. So the latency to the first spike, the duration of the first inner spike interval, the spike frequency adaptation over time. So all of those are features that we're trying to optimize with our fitting procedure, and then we end up with a model that hopefully will reproduce that. And so we've used these procedures to fit a variety of neurons. The data is still coming off the rig, so we're trying to fit those kind of as it comes. But you can see it's hard to see the morphologies, I guess, on the right, but basically we're fitting like big pyramidal cells at the top, fast spiking interneurons, as well as a variety of other types of cells. And you can see we're getting pretty good again fits to the overall kind of shape of the firing response, and in some cases pretty good fits even to the shape of the action potential. Now we are collecting a lot of more electrophysiological data than we're using to fit these models, right? We're doing more than just recording the responses to current steps. So we can use those other responses to test how our models are generalizing to those other types of stimuli. So here's an example of where we fit the model, again, based on the current steps, and then are applying a noisy current injection stimulus and comparing that to the experimental responses that were actually recorded. So at the bottom in gray is the current that was injected. The kind of darker shaded traces show the different experimental responses. You can see they are not completely consistent from trial to trial. And then you can see the model's response in red above that. And then the spike times are shown just for a little bit more clarity. And we can compare these to the experiment using a variety of kind of spike time metrics. So we're using this reliability metric on the right, basically saying, is the model that much different than the variability between the responses that you get from the single cell. And you can see from the plots on the right that the lines are pretty close to each other. So we're doing a pretty good job at capturing those spiking responses to 3 different random seeds of this noise stimuli. So we'd like to use these types of models not only for kind of defining cell types in addition to all this other data that we're collecting, but we'd also like to use this to build up these bigger models of cortical function, like I mentioned, that we'll then be able to compare to different experimental modalities that we're pursuing at the Institute, things like the 2-photon calcium imaging or extracellular electrophysiology recordings. So the strategy we're taking with the biophysical neurons is basically using this kind of library of single cell models that we're generating to populate a network, interconnected network of biophysically detailed neurons. So the example I'm showing is about 10,000 neurons representing essentially layer 4 of a small section of mouse V1. So we'll take these basically example models from our library. We'll connect them together using data taken from the literature as well as from in-house experiments. And then we're going to drive this network using basically visual input, but run through models being developed by a different group at the Institute, basically representing the LGN cells as LNP kind of filter models based on other data being collected at the Institute. So that'll generate kind of realistic spike trains that can be used as the feed-forward synaptic input to the network. And then we'll be able to test, basically look how the network is performing with a variety of visual input that we're applying to the mice in the awake case, and then even do perturbations in the network and compare them to similar perturbations done in experiments is the idea. So we're still in early stages of building these models. We're still just kind of showing proof of concept that we can tune them up and have them behave in a somewhat reasonable way. So here's an example of that biophysical model of layer 4 with feed-forward and recurrent connections. So you can see that we've been able to tune the synaptic weights. On the left, the firing rate, the average firing rate during the visual stimulus is shown on the y-axis on the left, and then it's just neuron ID at the bottom. So you can see the 8,000 pyramidal cells in this network are firing at kind of around 5 to 10 hertz, which is what we'd expect based on the experimental data. The fast spiking cells are firing a little bit faster than that. And you can see that we're generating kind of an irregular network activity closer to what we'd see. It's not an epileptic activity or highly synchronized or anything like that. So we can get the model to behave in a in a regime that we think is close to reality, but obviously this is very simplified, and we're still putting lots of data into it. So just to summarize, wanted to go kind of quickly just to as things are being shifted further back. But basically, I've talked about this high throughput experimental modeling effort that's underway at the Allen Institute, characterizing components of the visual cortical circuitry in the mouse. As I mentioned, these experimental data and models are going to be released to the public via our website, so look for that soon. We're developing these models at different levels of abstraction so that we can generate network models at different scales, including biophysically detailed ones, and that'll also let us see what the right level of abstraction or resolution is necessary to investigate or reproduce different cortical computations. And so our goals here are really to what the Allen Institute has always done, which is providing tools to the community to use, but also to develop a better understanding of these principles that might be underlying cortical computation, like what roles are different cell types playing in the circuit, how important is it to have different cell types participate in the circuit. So this is a picture of the Institute taken over the summer. We've added a lot of new faces even since this was taken. And thank you for your time and attention, and I'd be happy to take any questions.
So in an earlier part of your description of what you're doing, you pointed out that you and your colleagues are certainly yourselves aware that the dendrites have active channels.
For sure.
It's just that because of your workflow, you're only recording from the soma, so you don't have data up there. So I understand that already. But you probably also know that the response to synaptic input to the dendrites changes entirely if they're active. It's not just a small perturbation from a passive dendritic tree. It's profoundly different. It's nonlinear. Backpropagated action potentials have their effect on plasticity. There's NMDA spikes and sodium spikes and calcium spikes in dendrites.
Yep.
So, and I know that you and Christoph and everyone else there is fully aware of that. I just wonder what the roadmap is to address it.
Yeah, we're still figuring it out. So we do have people who are even working on building models with active conductances in the dendrites, even just based on the somatic data and making just some assumptions about how channels are distributed in the dendrites and seeing if we can fit those doing that. We're also thinking about different ways of potentially, doing some kind of directed dendritic input or even dendritic recording somehow, or collaborate with someone who's doing dendritic recording to kind of constrain those models better. So certainly something we're aware of, but yeah, we're figuring it out.
So there's a whole range of things you could do. I mean, if you had someone that could patch dendrites, that would be fantastic. You can provide synaptic stimulation to the dendrites to develop constraints on your model.
Absolutely.
You can also do immuno to get an idea of where the ion channels are of different types, and the Institute has deep capabilities along those lines.
Yep.
I'm sure you've talked about all those things.
Yeah, exactly. We're just trying to figure out what can be done more at scale. Yeah, things like dendritic recordings are tough to do that way. Yeah.
So my question has to do with kind of the motivation for such an endeavor, right? So I've always thought about it as being kind of like the massive physics simulations of nuclear explosions. So you do it so you don't have to do the actual explosions in order to know what happens, right? So is that kind of one of the motivations behind here? So we study the rat brain, so then we can do experiments on the rat without having to actually, work on the rat.
Not exactly. I think it's more just to have a framework to put different results from different modalities into. So it's, I think, like's been talked about at this workshop, sometimes it's really hard to integrate data collected in different ways through different modalities, especially when they're not done kind of in a consistent way or systematic way. So I think really we're seeing the models and simulations as an opportunity to bring a lot of data together and kind of force us to really think about what's going on all the time. We have the modelers and experimentalists working really closely together, so trying to feed off of each other, define new experiments and new directions. So it's really, I don't think it's to make actually doing the experiment obsolete. It's really just to try to continue to develop a deeper intuition into what's going on and generate testable hypotheses and kind of put all the data together in a consistent way.
And what about non-human primates? Why were rats chosen as opposed to...
mice, actually.
Mice.
Yeah, well, for the genetic tools. But we're also, a thing I did not talk about is that we are doing work in a similar vein on actually human cortical tissue. So neurosurgical samples, you can basically take those, slice those, and treat those as the same as a brain slice. They actually are better than mouse brain slices in lots of ways. So we're going to use the similar techniques, similar protocols to characterize those cells. We don't have genetic labels in that case, but still should be able to do some interesting stuff there. But yeah.
Thank you.